Good evening, everybody. I'm so happy to welcome you to the latest installment in the Torrey Botanical Society's Torrey Talks lecture series. My name is Catherine Mercier. I am the Society Programming Chair. I work for New York City Parks and also am finishing a PhD at the City University of New York. I'd like to give a little bit of information about the society before we start. So the Torrey Botanical Society was founded in 1867 and is an organization for people interested in plant life, including professional and amateur botanists, students, and anybody who simply enjoys nature. Today, the objectives of the society are to promote an interest in botany and to collect and disseminate information on all phases of plant science. These objectives are fulfilled through meetings, field trips, lectures such as these, publications, funding of graduate student research and education, and sponsorship of symposia and regional conferences. The best way to stay updated on everything that we're doing, future lectures, field trips, and other activities is to become a member, which you can do on our website, www.torreybotanical.org. You can also follow us on social media. All of our handles are up on the slide. We are at Torrey Botanical almost everywhere on Twitter or X, Facebook, Instagram, and most recently Blue Sky. And then we are at Torrey Botanical Society on YouTube, where you can find recordings of all of these lectures. So before we begin the talk, just a few notes about Zoom. Please do keep yourself muted during the talk, but you're welcome to have your camera on or off. You can enter comments and questions in the chat as we go, as the talk proceeds, but we'll really address those during the Q&A portion of the talk after the lecture. At that time, you can either use the raise hand feature to raise your hand and ask your question aloud, or I can read your question from the chat if you'd prefer. And if you'd like to share the talk with someone else or simply rewatch it later, it will be recorded and available on the YouTube page. So tonight's talk will be presented by Emma Fryer. Emma was originally invited as part of our series last semester, so she was awarded a Torrey Graduate Student Research Fellowship in 2021 while working on her master's thesis at Cal Poly San Luis Obispo, which I hope I pronounced correctly. While she was there, she was working with a suite of annual wildflowers native to the San Joaquin Desert of California, which you can see on this title slide here. These annual wildflowers are among those that create the superblooms that have recently attracted international attention to an otherwise overlooked and imperiled ecosystem. The western edge of the San Joaquin Desert hosts two of the last remaining tracts of intact habitat in the region, and both of these sites host an unusually high number of rare and endangered plant and animal species. So before I give too much away, I will hand it over to Emma to talk to us about anatomy of a superbloom, soil, competition, and survival in the San Joaquin Desert.
I first, if you're not already familiar with it, wanted to explain the San Joaquin Desert. It was only described in 2011, in part because the landscape in this area has been altered really dramatically over the last 200 years. And so it's just this area and this polygon here. And for most of the year, it looks like this. So you would think it does certainly look like a desert. You would have expected that it would have been described as one a bit sooner. But before 2011, this region was actually usually described as a grassland, and it was kind of assumed that it had been dominated by native bunch grasses prior to the arrival of invasive grasses like Bromus magitensis. And while these invasive annual grasses certainly do dominate the landscape today, it turns out that that interpretation was based on one man's description of a single site, actually my hometown. And it was basically one citation that spiraled out of control, combined with the fact that today you do see invasive annual grasses all over. And it's kind of hard to imagine what it used to look like today. The paper that first outlined this region as being a desert noted that nearly 60% of it has been like permanently altered by human activity. And On the floor of the Central Valley, that number is closer to 97%. And if you look at the region now, you really wouldn't even see enough unaltered land to consider what it looked like unless you get pretty well outside of the valley floor. And so when I first came back home from college and I started working in the region as a consulting botanist, I kind of started to learn about all of this. And not just how little we knew about the prior landscape, but also the number of threatened and endangered species and what that meant, which was basically that we'd lost quite a lot before we knew what it was. And I started trying to learn about these species that used to be here and trying to learn about all of the rare species that still existed. And I couldn't find a whole lot. And that was around the time that this image circulated online. And can you see my cursor? Hopefully.
Yes.
Thank you, whoever spoke up. And this is a simulated image, but this shows a pretty accurate interpretation of what California might have looked like before Europeans arrived and the region was really transformed by agriculture, development, and oil extraction. And while this is true for a lot of places, it really stuck with me and ultimately actually led me to what became my thesis project. And I'm going to fix this because you were not seeing the whole slide. Pardon me, everyone. So for most Californians who do know this area, this is just kind of like a brown, dry expanse. And it's basically what you pass through when you're going from LA to San Francisco. And it's often the butt of jokes. And that's honestly kind of the message you get growing up in the region. And it does look truly pretty desolate and brown for most of the year. But in good rain years, we get this really magnificent transformation. And it's absolutely striking to see in person. This is one of those things where photos don't do it justice. And in places that are relatively well preserved, like here, Carrizo Plain, it's a really, really incredible sight. And if we look at historical accounts of the region, you can actually find accounts of pretty much the same impression as far back as the 1800s. The first description of the region that I came across was from John Muir, when he described it as like a lake of pure sunshine, 40 or 50 miles wide, 500 miles long, one rich furred garden of yellow compositae. And as the floweriest piece of world I ever walked, one vast, level, even flower bed, a sheet of flowers. And 20 years prior, another man described it as like a crazy quilt of color, where the quilt was flowers, rows and rows of rose, yellow, scarlet, orange, and blue, each species attached to a different soil type with some colored bands stretching a mile across. And my mother cried with joy and wanted to make a home right there in the middle of it all. And as recently as like 1942, you can actually still see this in a lot of the region. This photo was outside Bakersfield in the 40s. This is the same general area in the 30s. And this is the oldest photo I'm aware of, not in color, so it's hard to tell that those are flowers. But today, these locations are dominated by invasive annual grasses or have been actually fully developed or cultivated for agriculture. But there are two places with relatively well-preserved communities of these wildflowers. And one of those is Cantua Creek, which is located here, and then Carrizo Plain, which I already mentioned here. And so these were the study sites for my master's thesis, and I'll give a bit more background on them shortly, but I just want to share a little bit of what this region hosts, because these places do still preserve quite a lot. Carrizo Plain is a hot spot of endangered and threatened species, and after a good rain year, both of these places, my study sites, are really overflowing with an incredible flora. And this is just stuff that I had photos of. Visiting them is a little bit like feeling like you stumbled across some kind of hidden treasure. You'd have no idea from the interstate that this is so close by, unless you went looking for it. And it's not just plants. These places are hosting numerous endangered animal species. This scorpion was only recently described, and it's only found in Carrizo Plain. Even in dry years, when it looks quite barren, there's a really remarkable lichen flora on the plain. And this is a photo of some UV fluorescent flora lichens making up a crust. on the soil from a side project I did with some undergrads. And we actually found 2 lichens that it turns out are new species that someone else is describing. And so despite kind of barren appearance for most of this year, this region has a lot for us to see and a lot for us still to learn about. And kind of coming back to these study sites, they were chosen not just because of their comparatively intact state, but also because they have really unique soils. And this is already a desert, right? So it's arid, it's hot. By default, it's difficult for plants. But my study species at my sites are also growing on vertic clay soils, which are on their own pretty stressful for plants. And if you haven't heard of them before, that's fair because they're pretty uncommon. These soils have really high clay content. Specifically, they have a minimum of 20% smectite clay, which means that they absorb a ton of water relative to like kaolinite. And so this means they expand a lot when they're wet, they shrink back down a lot when they're dry, and it creates this very distinctive cracking when it dries. But that cracking is also creating physical stress because the soil will contract as it dries and the cracks form, which can actually physically tear plant roots. And then high clay content of any kind creates water stress for plants because clay particles are really small. This is a smectite particle. That means that the space in between them is going to be smaller. And so that means the water tension of the water in between those particles is smaller, which ultimately means that very high clay soils can have lower water potential than plant roots and can actually keep the plants from taking water up from that soil pore space, even though the root has access. And it gets worse. These soils are also frequently high in sodium. Because both my study sites are in geologic units of marine origin, their soils have high sodium that's inherited from marine shale. And if that clay runs off into basins like it does at Carrizo Plain, dissolved salts can accumulate over time, which you can see at Soda Lake. And so these are overall just really extreme soils with several sources of stress for plants. And so I focused on species that can grow on these soils, but more than that, I focused on species that we had reason to believe were mainly growing on these soils. And so I actually looked at California herbarium records to see what percentage of records, of all the records where they mentioned substrate, were actually on clay soils to confirm this. And I did find that those records reflected strong affinity for vertic clay soils for these species. So we've established that we have these really stressful soils, and we've established that we have edaphic affinity present in this community. And lastly, and maybe like the coolest, most glamorous part, is that these sites have these species growing in these really distinctive mosaic patch-like patterns that you're seeing here and here. And one of my committee members, Ryan O'Dell, is a botanist for the region, and I had the benefit of his experience with the system already. And it did look likely, based on some preliminary soil sampling, that these patches were actually reflecting differences in the soils, and that these soils were as heterogeneous as that mosaic pattern of blooms suggested. And of course, I should note here, aspect of and slope of hills is probably a factor, but for this site, I actually focused on the soils in this foreground area here on the floor to focus on the soil properties. But if we look back at those quotes I brought up earlier, this observation about soils and the dominance of a single species and on a single soil type has actually already been made before, but no one's ever looked at it. So we have this combination of all of these other cool factors with edaphic heterogeneity going on. And this is interesting because heterogeneous landscapes kind of by default entail a higher number of ecological niches. And by niche, I mean the biotic and abiotic conditions those species can survive and persist under. So a species is going to have some specific range of environmental factors under which it can survive. And so let's say for this species, of all these possible niches, so all these colors in the landscape here, let's say it's only capable of surviving on these soils in the foreground. So just these soils, and that would be the species' fundamental niche. So these are the conditions under which it is capable of surviving and persisting. But in an actual community, actually growing out in the world, there's going to be other factors like coexistence and competition. And so it's going to affect its ability to actually survive on these soils when competition is around, which is going to narrow that fundamental niche down to a realized niche. So it can survive and persist on these soils when competition is present. And so that realized niche would be the one we actually see plants in when we see them out in the wild. And so that's basically what we thought that these mosaic bloom patterns were, that they were a reflection of these species' realized edaphic niche. And so if that's what's going on, it's super cool, and there's a bunch of cool questions we can ask, but no work had really been done on this system, so I wanted to establish this fact first. If these patches reflect realized edaphic niches, we'd expect to see differences in the soil between those patches. And so my first component of this project was a field survey effort to try and establish that this is what we thought it was. And then I followed up with a greenhouse study, rather a pot study, because it wasn't inside a greenhouse, to answer additional questions. So for the field component, I actually went out to my sites and I found these patches where we had a single species clearly dominant, and I collected soil samples from the centroid of each of those patches. For each species, I did this for multiple distinct physically separate patches for at least 5 patches. And once I'd done this for all 12 study species, I had the soil samples tested. And I did this for several properties that are physiologically relevant to plants, those that we thought were most likely to be affecting the plant on like a very basic level or acting as stressors. And to clarify, CEC here is cation exchange capacity, so the ability of the soil to hold a positively charged ion, so that's affecting mineral uptake. And then coal down here is coefficient of linear extension, which is a measure of that shrink swell I was talking about earlier. And so basically what we did was we would just saturate the soil, extrude it in this rod, and then measure its length, let it dry, and then measure how much it decreased in length to see how much of that physical stressor was present in those plant roots. And so since all these properties can affect plant growth, and there's just a lot going on with soils in general that can affect plants, I really wanted to capture really as much information as possible about them. So I did a principal component analysis for these soils with all of the results. And that's basically just taking all of the variation in these soils and distilling it down to 2 axes, these principal components. And so those points that you're seeing are all of the soils from the species patches plotted along those 2 axes that explain the variation we see. And so the total space is basically like all possible types of soil and where these individual samples fell within them. And so these principal components are defined by specific properties more than others. So in this case, we have phosphorus, sodium, CEC, and the ratio of sodium to potassium. And so soils moving this way are going to be higher in those. The second one was mostly defined by calcium, magnesium, and the ratio of the 2. And so I feel like I have to mention at this point, if you were wondering how my study sites compared because they were physically disjunct, I did look into this and I plotted 95% confidence ellipses over these points, separated by which site the soil was collected from. And purple and blue is probably a bad choice because the overlap is slightly less obvious, but we did see some overlap on both our principal components. And so we saw that in both cases for both those sets of properties that were really defining the differences in these soils. And then if we look at these points distinguished by species, so which species patch the soils were sampled from, it's kind of a jumble because it turns out that 12 is maybe too many species to have in one plot. But if you actually like look at it species by species, we have Lepidium druidii, subspecies druidii here, one of my favorites. And it's extending way further out here than these other ones into soils much higher in these properties here. And in this case, it's because this one is growing on soils with just crazy high sodium, like 15,000 parts per million in those soil samples. And if we look at the other subspecies, Lepidium druidii subspecies album, it's actually in a pretty different part of our soil space, right? It's much narrower in terms of this axis than the range of the other subspecies, and the same on this axis. We've got a much broader range here in terms of these properties relative to that other subspecies. And then some species like Leptosne Calliopsidea had much, much narrower range of soils that they occurred on within this space. And some species overlapped, others didn't. And so what we're seeing when we look at this is the apparent realized edaphic niche for these species and how they compare to one another. But it's kind of tricky to make any conclusions with this plot. 12 species is just like too much to eyeball. And so what I did was I took where each of these species soil samples fell on each principal component, so like the actual like coordinate of where it fell. And I did this so that I could compare the species in terms of those coordinates. And so this is all of these species soils plotted by where they fell on that principal component. And I did actually see a difference between the means for these species points on the axis, which would translate to a difference in niche as defined by these properties. And I also actually saw a difference in variance, the spread, which would suggest that these species also have differences in the breadth of their niche, how wide a range of soils they occur on. And I did the same for the other principal component, which you'll recall was defined more by different properties. And we still saw differences in the mean, so differences in their edaphic niche defined by those properties. And we saw differences in the breadth of that niche. So it does look like soils are different between these species patches, and that suggests that these species have different realized edaphic niches and that those niches have different breadth. And the other part of why this system's heterogeneity was so interesting is that, speaking generally, an environment with low heterogeneity of abiotic factors, so say just 2 soil types, is going to have fewer niches available for species to occupy. And with greater environmental heterogeneity, you're going to have a higher number of niches available, which means potentially more species and greater diversity. But that also means that communities respond to things like invasion differently. And so that's kind of where the second part of my master's thesis comes in. Having established we have these species and these very visible realized niches, which on its own is really cool, on this really stressful substrate that's very heterogeneous, I wanted to know what their fundamental niche looked like. So what kind of soils are these species capable of surviving on without competition and coexistence present? At least in terms of the soil across, at least in terms of the soil and across the gradient of edaphic stress that's present at my sites. And also, Bromus madridensis is a major issue for most of California, and I wanted to see if it affected their performance. I had a good guess, but I still wanted to check. But then also, how does competition affect their niche optimum? What is competition actually doing in the field at these sites? And so, oh my gosh, did I lose a slide? Okay, sorry. Based on the fieldwork, I was also really interested in seeing how competition affects these species given the stressful soils, because there's this idea in like, and I don't have like a less snooty sounding way to say this, but an idea in contemporary niche theory that the outcome of 2 species competing, and I apologize, I have a really great animation here that none of you are seeing. But the idea is that if you have 2 species competing under an environmental stressor like a harsh soil, it's predicted that the outcome depends on the species' tolerance of that stressor, where ultimately the species that has a higher tolerance for the stressor is going to eventually fully outcompete the other. And all of these dots are one species that's outcompeted another on a stressful soil, if my animations were working. So I wanted to see if that was happening here and if this was contributing to these single species patches. So I did a pot study with the same species that I did all of that soil sampling for in the field. And I wanted to see the effects of soil and competition on these species, but in an experimental manner. And so for the treatment soils, I needed to try and get like a gradient of the edaphic stress, the soil stress at these sites. And if we look back at the PCA and what really defined them, it was these properties here that really determined what the variation in these soils was. And so I focused on these properties for my pot study, and I got 3 soils collected from near my study sites, which cover a gradient of those properties. And you can see it worked out very nicely that I got a pretty good gradient across all of these major things in terms of sodium, the sodium to potassium ratio, which covers sodium and potassium, calcium to magnesium ratio, and that soil cation exchange capacity. And so what I did was I planted each species across each of these soils, and I have them referred to by sodicity here just for like ease of talking about it. And I repeated this both with and without a competition treatment. And so that competition treatment consisted of 3 Bromus madridensis plants seeded with the native species, and then I weeded it down so it was 1 native species with 3 Bromus plants. And then I did this with 4 replicates of each treatment combination. And I did this for all 12 species from seed to seed set over about 10 months in 2021. So this was like what I was doing during COVID. And it was this really big undertaking, in part because these soils are so unusual. You can't just water the soil from the top of the pot and expect the water to go all the way down. What would happen was it would like permeate the first inch or two, and then the whole pot would like stop getting water. It would just make a seal. And so I had to like manually wet the soils by hand for 288 pots, seeded them, and then I had to weed out all of the volunteers from the seed bank. This is a really great time-lapse video of me doing that, which you cannot see. And it actually went really well. It was a lot of work, but it went like shockingly well. Germination was great. I lost hardly any replicates, never more than one. And well, I'd originally planned to look at root biomass, shoot biomass, and reproductive biomass separately. They were all ultimately telling the same story. So I'm focusing today on total biomass. And so what did the biomass tell us from the study? All of these bags here are not even all of the biomass. It's quite a lot. And I have this here to give credit to my undergraduate volunteers who were amazing. But for the sake of time, I'm not going to really dig into the analysis details, but instead kind of talk about the results. So first question, did we see a niche optimum between the treatment soils that suggested a fundamental niche exists? At the field sites, we saw that there was preference for some soils, and I confirmed with herbarium data that this was generally true for these species. But if we have no competition in the picture, is it still true? And so this is just the treatment that had no competition. I'm doing just one because 12 box plots is too many. But looking at Venetoa occidentalis, for this species, we did see that biomass was significantly higher on this soil relative to these other two. So we did see some kind of edaphic niche optimum existed across the soils for this species. And these treatment soils might not have caught the true niche optimum for this species in terms of the soil. We have evidence that one exists based on these treatment soils that I used. And please do not try to read every single plot in this 12 plot slide, but this is just so you can see the general pattern that this was the case for all of these species. So this is a pattern in this community that we saw higher biomass on at least one soil relative to at least one other when there was no competition present. So it does appear that these species have some kind of fundamental edaphic niche. At the very least, we're saying that niche optima exists just by the soil when competition isn't present. And what about when competition was present? First, did Bromus magitensis being present reduce biomass? And so lighter gray in this is without competition, or with competition, darker gray is without. And don't try to read all of them, but just note that the competition treatment, it was universal. Biomass was really reduced for all of the species, except in one case where did not technically meet our significance threshold. Those of you who care about this part, the interaction was significant though, so it was kind of a non-issue. It was technically significant because the interaction was. And so we see a really strong overall pattern, right? That competition from Bromus magitensis is affecting these species' performance. And I mean, not entirely surprising based on what I told you earlier about it, but the cool part is that we saw that these species had a fundamental niche in terms of soil, or at least evidence that one existed. And when we add in competition, does the realized niche exist like we saw in the field, or is it just the same overall? And then also, did competition affect that edaphic niche? And if we just focus on Phacelia ciliata for simplicity, if we look at competition's effect on biomass, so like the difference between the competition and non-competition treatments, we actually saw a difference in how much competition affected it depending on which soil it was on. And so without competition, the soil niche optimum for Phacelia ciliata was this soil here. But when we had competition present, it actually changed to a different soil for the niche optimum. And so we actually saw this in several species, and it was significant for the community overall. And so these species' competitive ability actually differs within the range of soil chemistry captured by my treatment soils. And it looks like competition can actually cause shifts in edaphic niche optimum. So like the best soil for these species was different if competition was around. And so when you look at this really striking patchwork of blooms, these species patches that we're seeing do reflect differences in the soil. These species do seem to have realized edaphic niches that are distinct and have different breadth. And we saw that there's evidence of fundamental edaphic niche for these species. Bromus madridensis, perhaps surprising no one, negatively affected their performance. And most interestingly, competition can cause shifts in these species' niche optima. We can actually see a change between the fundamental and realized niche when competition comes into play, or because of competition, excuse me. And so this last part is especially interesting to me in what it suggests. If species can do better on a different soil when competition is present, again, missing out on a really great animation here. If you've got all this, I'll do my best without it. If you've got all this local scale heterogeneity, that means we have more available niches, then these species have more options in terms of soil. And so if we have some species that has a fundamental niche optimum that's, say it does best on this brown soil, which we'll say is low stress, but it can also, that its fundamental niche includes this gray soil that we'll say is higher stress, then if this species encounters a competitor, this green one that can outcompete it on the brown soil that's lower stress, that species can persist by having a realized niche optimum on this higher stress soil. And ultimately, you'll see them get kind of like separated into different realized niches. But in this scenario, that species that had that broad fundamental niche has been able to survive on a sort of refugium of that high stress soil. And so my conclusions from this master's project overall was that it does look like a combination of soil heterogeneity and these really extreme soils is part of why these species have been able to persist through this really aggressive invasive annual grass. And it looks like that's part of what's behind these really dramatic mosaic pattern of blooms. I feel like I have to say at some point that like geology is obviously a factor there as well. To be clear that my results are consistent with this, but they don't prove this. I had limited soils in this study, and this was a master's project with master's project limitations. And it would take a population level set of metrics to really show that this is happening. And unfortunately, those clay soils are so extreme that it is physically impossible to drive on them if they're even a little wet. So I don't know how likely it is this system will be the one that shows us this, but it's very interesting to think about. And just to give it a bit more context, what does this dynamic between these extreme soils and the effect that competition had on those niche optima mean for these species survival? Because this is something that's been on my mind the whole time I've been working with these species. And I was kind of surprised that I feel good about the outcome, because there's been some other work recently that found that some species similar to those in my study that have really, really narrow realized edaphic niches were like found on one soil type only. For some of those species, it actually looks like their fundamental niche is only narrow in like that one axis of the niche, right? Only in terms of soil. And so as climate change progresses, just because we see a really narrow realized niche, it doesn't necessarily mean that we're going to see all of these highly endemic species disappear if they have a broader fundamental niche that we're simply not seeing, or if they just have a broader niche in terms of other factors besides that one that they're really restricted with. And so if climate change or more invasive species alters the competition that these kinds of species are facing, we might instead see a shift in realized niche to a different soil type or a different microclimate. And also, others have found that many of these invasive annual grasses, which have really transformed the region, don't have the same capacity for deep physiological seed dormancy that the native annuals like my study species out there do. And so it's also possible that as climate change progresses and this region is expected to get longer, more severe dry periods between good rain years, we may see these species survive longer in the seed bank than those invasive annual grass competitor seeds can, and potentially survive in a different realized niche that way as a result. And so I was definitely a little surprised to feel like, okay, like a little optimistic about this system, but I just wanted to add that in since this is a little bit more casual a talk. And I was very pleased with that outcome or that possible implication. I want to say thank you for your attention and your time, and also just like the Torrey Botanical Society support was so important, I basically self-funded this project. And being in a well-funded institution now, that means like twice as much to me now. Torrey Botanical Society was one of several very generous organizations that made it possible to pull this project off. And just so many people. So thank you all. I apologize for the lack of animations and difficulty with the slides. Any questions?
Thank you so much, Emma. That was a beautiful talk. If anybody has any questions, feel free to throw them in the chat and I'll read them. Use the raise hand feature or go ahead and unmute yourself if you would like to ask a question. I will jump in quickly. I'm curious if you are like, is any of this work sort of inspiring or building into what you're working on for the PhD?
I'm not gonna lie, no.
Okay.
Well, I've actually been exploring ways to continue work with the system in a postdoc. There's an interesting Swiss fellowship that citizenship does not matter for, which could potentially send me abroad on Swiss money. And so I've been exploring whether or not I could work with this system again. I did end up doing a side project working with the lichens in Carrizo Plain, which was really fun. And I have a side project looking at clay endemism based on herbarium records that kind of stemmed from me checking these species initially. So I definitely still have ongoing work. My PhD will actually be focusing on anatomical and physiological traits of seedlings and how that affects forest regeneration.
Oh, cool. Okay.
I swear the animations were really good, you guys. I'm super bummed. Thank you to the chat. The photo from the 1800s, where did I find it? That came from a historical society in, it was like Tulare or Kern County, like the non-governmental organization type of historical society. They had online records. And I think also it was in the, like, it was like Boston University or Harvard or something. They had excuse me, 1 a.m. here, digitized records from a like railroad expedition to scope out rail lines. And like this guy was just like taking pictures of people doing things, and it ended up on like the Google historical documents thing.
Cool. I'm curious if like your data has this ability to see, like, were the conditions that the species liked in the pots, like their optimum in the pot, did that match with the soil that they were found on in the...
I tried so hard to find a way to do that. I tried so hard. I was so sad. If I did it again, I would have physically taken their soil from their patch to do it. But you know, rare species and also remote location. But I didn't ever find a good way to do it. I honestly couldn't give you a good answer. Like it just genuinely, I couldn't find a sound method of doing it. I've still been pestering a couple postdocs here about it, but I think I may never have a great answer for that. And it's definitely one of those things where it's like you pick your PCA and your answer differs based on the PCA. It was very frustrating because I really wanted to answer that question. Chat questions. What encouraged me to continue the experiment despite having to manually water 200+ pots? I mean, I feel like it was probably clear at the start of my presentation that I care a lot about the valley. I grew up in a very unglamorous Fresno, California, and to me, this system is a little emblematic of the region. It's very economically and environmentally impoverished, and it felt important to me to learn about it and learn about the place I grew up because I had never learned any of like the local natural history until I came back after college. And probably it helped that COVID was going on and I was just like in my friend's backyard 24/7.
Before we go to the next chat question, Frosty, do you want to? Yeah. Are you able to unmute now? Frosty, I see your hand and it looks like you're unmuted. Did you have a question? Okay, while we wait, Michelle asks, there's going to be quite a bit of restoration work happening in the SJ Valley. Do you see any lessons from your study that could inform the restoration work happening?
So I did actually work as a consulting botanist in the region. And I mean, I think for one thing, there is not there's not enough published work on like the very basics of these species. It feels like you're really scrounging for enough information to make like good environmental planning decisions for them. And so I would say the first thing is that like people need to get the plant press out of their trunk and submit the records with their notes. And I say that with a plant press in my trunk, but I think very, very much accounting for the soils. I feel like a lot of that region, I was seeing projects where they were just reseeding and like doing the hydro spray thing after, and it was not accounting for the fact that like when you have these really crazy harsh soils and like you're in this region where like the soil being too salty to grow stuff is a problem. Like if you have really extreme conditions like that, you need to be accounting for them when you're doing restoration planning or like trying to mitigate. But I do also suspect that there are some cases where species are basically hiding in the seed bank as refugia and then showing up when conditions are good. And I have like a sneaking suspicion that there's like a few rare species that are maybe like not as rare as we think because they're just like waiting for the right amount of rain. That's based off like, like one really good bloom year in Carrizo.
Okay, Molly asks, do any of these study taxa ever show up, say, on, say, highway roadsides?
Yes. Okay. So, well, for sure, yes, one way. That's kind of cool. So the Coast Range on the west, wow, I had to think about that. On the west side of the valley, there's whenever we have really heavy rains, for one thing, that portion of the valley is currently flooded. Tulare Lake is back right now, which is astonishing. But that Coast Range stretch that falls within the San Joaquin has 0 trees. And those soils will wash out because that area is so arid that it's just like not getting fully eroded ever. And so when we get really heavy rains, like all of that soil washes out with it. And there's all of these spots along I-5 with these like underpasses just for these arroyos. And they will dump so much soil from the Coast Range that you will randomly get species growing under and around I-5. They're like rare species from the Temblor Range for like a year or two. I mean, they're waifs, at least for now they are. I mean, maybe in the future that will change, but they're like waif populations that just show up for the year or two after a good rain year. Like I'm sure right now there's like Colanthus and Flatus on I-5 and someone somewhere is very confused about it. as far as like roadsides without a weird situation, Leptosne Caliopsidea is probably one of the more generalist of them, and I am guessing is on plenty of roadsides throughout the region. All of them are pretty specific to the San Joaquin Desert and very much associated with the Coast Range. Monolopia stricta and the other Monolopia also probably, but they are definitely not the rarest ones. I do feel like the rare ones are really only showing up on roadsides, like at the sites.
Okay, Frosty's question. Sorry, we couldn't make the mic work, but I see you put it in the chat. And I was also thinking a similar thing. So Frosty asks, can you speculate on the competition result if you had used other native species rather than the bromus?
I do think that using the annual grasses adds this extra like phenological complication, right? Because like a big factor in like whether or not we see good bloom years is like the timing of the rain, because these annual grasses sometimes are just like too fast, right?
Mm-hmm.
And so I suspect that that was a factor. Also, that some of my species did seem like they bloomed late enough that they kind of sidestepped the competition. I have a massive phenology data set that I should not have collected that I have not used yet that I'm hoping to eventually publish. But the stat, I need more time on the stats for that. My suspicion, and I wish that I had done like each of these species against each of the others, is that you would see very noticeable, or at least like not insignificant patterns where you saw like the amount that the competition from another affected it was absolutely related to how well that species did on the soil. And you'd kind of see like what I was talking about happening in a scenario of like high stress environment, two species competing, where you would see one outcompeting the other like until it was completely eliminated from that niche. And I would expect to see that the outcome of native competition would reflect that, and maybe even more clearly than we saw with the bromus, because again, Bromus magitensis and a lot of these invasive annuals are just like a little phenologically disjunct depending on rainfall.
Thank you. Very cool. Any last questions? Okay.
Well, thank you everyone for the audience.
I like to end all of the lectures with this final question, which is, if there is like one takeaway, one thing that you wanted the audience to leave with, or one takeaway from the talk, one lesson, what would it be? You can take a minute to think about it if you'd like.
I think that species found on harsh and especially heterogeneous harsh substrates or in harsh environments are probably a little tougher than we give them credit for. And that I think we maybe need to be a little less laser focused on negative outcomes with them when we're projecting for climate change. Admittedly, my current work is like going to inform climate change models for forests, so I might also be like a little biasing myself there. But for me, it was kind of a shock to realize that like what I found suggested something very different than what I had assumed about the system. And I think it just makes all of these species that are found on these crazy substrates like 10 times cooler that they're doing it, and like they might actually just keep doing it as like climate change progresses, which I don't think I'm always an optimist, but I think it's something to bear in mind because it can be a hard field to work in, especially if you're doing like consulting and planning.
That's awesome. Thank you so much. Thank you for being here. Thank you for talking to the society. Thank you to everyone in the audience for being here today.
Yeah, thank you everyone for having me.
Yeah, so we'll have our next story talk in March. Stay tuned for announcements and become a member, torreybotanical.org. All right, have a good night, everyone.